Hello everyone, this is our project submission to the problem statement identifying taxonomy and accessing biodiversity using eDNA dataset. So the problem we are trying to solve here is the problem of analyzing eDNA and classifying into various taxonomies. See, when we collect eDNAs from the sea or various kind of environments, we don't have a fixed kind of data set to compare it against. Like deep sea is highly unexplored, so there is incomplete and sparse database which cannot be used to compare against the DNA samples that we have collected. So it creates a kind of gap in knowledge. So what we are proposing to solve this solution is an AI-driven deep learning pipeline, which will take raw DNA as an input and will use it on fine-tuned higher DNA, that is attention-free sequence model. Like it is based on a Mamba architecture, and it is the SSN model. It is a state-of-the-art model, and we'll combine it with the environmental metadata by giving it an extra layer, and it will generate an embedding. We'll plot that embedding in 3D with the help of UMAP, and then we'll run some clustering algorithms like HBD scans to make clusters. So what our proposed solution gives uniquely is that we treat DNA as a biological language. We'll fine-tune higher DNA on an attention-free. It is an attention-free long sequence model, highly efficient, and on the large eDNA corpus. And we'll also use an additional environmental metadata layer for robust classification. So what is unique is that we are not using a transformer architecture. Unlike transformer, this higher DNA has a capability to sequence long sequences genomics. Unlike transformer, it is computationally inexpensive to train. Like the transformer architecture runs on big O of L square time complexity, but this model will run on O L log n complexity. See, then we'll do the unsupervised biodiversity mapping. Like we'll apply the UMAP and HBD scan clustering to embed, uncover hidden taxonomics dependencies. Like when we cluster different things, like we'll get to know about different relations between different taxonomies which were earlier unrelated or unknown to the humans. And see, Mamba selective state open space model is very like hyper efficient. Unlike transformer, it does not work on the attention seeking parameters, but on a different parameter which is easy to train and use. So what our architecture rule looks like is we give the raw DNA input to the model, its sequence processing, then encoding as nucleotide tokens. Nucleotide tokens is the token system that was used in the nucleotide transformers. We'll encode as the nucleotide tokens. Then we'll take that token generations and give it to the higher DNA fine tuned. I'll show you just in a moment how we fine tune the model, higher DNA model. That is, then we'll take the latent space embeddings, all the embeddings from the thing, and give it to the environmental metadata layer, pass it through the metadata layer, and then we'll dimensionality reduce, we'll use UMAP, then unsupervised clustering for clustering different things, and then we can get the insights. This picture is a description of the higher DNA model. What we'll do is we'll use the same model, we'll freeze all the this pre-training operators and parameters. What we'll do is we'll introduce new layers for further classification, for further downstream tasks. So a proof of concept, I will show you now what we have implemented and the proof of concept. See, in this notebook we have tried to fine tune the model on the data set. That is, we have fine tuned on this data set, that is nucleotide transformer downstream task. This is a data set by InstaDeep AI. What we have tried to do is we have implemented this in this notebook. See, we have loaded the data set and everything. What we have done is we have the precision metrics, like it is very good in evaluation metrics, all have given a very strong results, and this the confusion matrix also shows that the model is quite confident. We have also prepared a Streamlit dashboard to show you a kind of DNA classifier prototype. We have a DNA. What we try to classify this particular thing, we have trained for the sequence. Tata sequence is like a thing present in eukaryotics that gives generation sequences. So this is the sequence. We'll click on predict. So it will run the model that we have just fine tuned. We have just fine tuned out here, and use that model using Streamlit and give you a sequence. Like it has predicted class 1. Class 1 is actually an eukaryotic cell. We have fine tuned this model on a very small scale currently. So that's why there are only two classes, two classes. So the sequence, you can see the model probabilities here. So this kind of establishes that the Mamba sequence can be used for DNA classifications and different taxonomies identification and all. And it is a very strong model. Unlike a transformer, it is computationally inexpensive and efficient, and it can give a very strong results if trained on high scale with all the data that is available. This can be a proof of how DNA can be analyzed further down the line. Thank you.